Welcome to our lesson on the food test practicals. The key questions for today are 1. How is the test for the biological molecule starch carried out? 2. What reagent is used to test for the presence of sugars? 3. How can we test for proteins? 4. How do we test for the presence of lipids? 5. How can these tests be used to determine enzyme activity? The key words you will need to know are starch, glucose, Benedict's solution, Baccalaureate reagent, iodine. The presence of biological molecules can be tested for using different qualitative reagents. This means that we can test for the presence of the molecules, but we will get a qualitative result and not a quantitative one, so we cannot draw conclusions about the amount of the molecule present. Task: name 3 different biological molecules. The different biological molecules are carbohydrates, proteins, and lipids. The first tests we will explore are the tests for carbohydrate molecules. Starch is a complex carbohydrate made up of many glucose molecules joined together. Glucose is a simple carbohydrate made of single molecules. Both of these are carbohydrates, but the tests are different. Starch is tested for using iodine solution. Iodine solution is orange in colour and it turns blue-black in the presence of starch. This means a positive result for the presence of starch is a colour change from orange to blue-black. A negative result for the presence of starch is the iodine solution remaining orange. Food items such as bread, rice and potatoes would obtain a positive result for the presence of starch. The test for reducing sugars, such as glucose, is done using Benedict's solution. Benedict's solution must be mixed with the sample and then heated for several minutes in a water bath. Benedict's solution is bright blue and will turn green, yellow, orange or brick red if glucose is present. Green indicates a small amount of glucose and brick red a large amount. Any of these colours represents a positive result for the presence of glucose. A negative result would be if the Benedict's solution remained blue. Glucose solution or sugary sweets would give a positive test for glucose using Benedict's solution. We can test for the presence of protein using Baccalaureate reagent. This is a blue solution that turns lilac when protein is present. This is a positive result. A negative result is when the Baccalaureate reagent remains blue. Meat, egg white and protein powder would give a positive result for the Baccalaureate test for protein. The test for lipids has 2 steps. The first is to add ethanol to the sample, then add an equal amount of distilled water and shake it. If the solution remains clear, then no lipid is present. But if it goes cloudy, this indicates that an emulsion is formed. An emulsion is a positive result for the test for lipids, indicating lipids are present. Oil or butter would give a positive result for the emulsion test. Task: make a table of the different tests and the results for each. Extension. How could these tests be used to test the rate of digestion of these molecules? The food tests for starch, protein, and lipids can be used to test a sample periodically to check for the presence of the molecule. When there is no longer any of the molecule left, then we know it has been fully digested. This means that we can test food samples with enzymes at different temperatures or pHs and see in which condition the molecules are digested the quickest. Temperature can be changed using a water bath, and pH can be altered using a pH buffer solution. An example of a practical using this method is to test amylase activity at different pHs. The amylase and 5 cm cubed of starch will be mixed with different pH buffer solutions to make different pHs. For example, a pH of 4, 5, 6, 7 and 8. A sample would be removed every 30 seconds and tested with iodine on a spotting tile. Once the starch is digested, the iodine will no longer change colour. If this occurred after 120 seconds, then this means that 5 centimetres cubed of starch was digested in 90 seconds. This gives the following rate of digestion of starch. Task. Complete the following AQA exam question. A student investigated the effect of temperature on the activity of human amylase. This is the method used, as follows. Why did the student leave the starch and amylase solutions in the water bath for 5 minutes in step 3? The student left the starch and amylase in the water bath for 5 minutes so that they were both at the desired temperature before they were mixed. Extension. Answer the following AQA exam question.